Thank you very much, everybody, for joining. I'm very pleased to welcome Professor Matt Jones from Bristol to speak to us about sleep and something of interest to us all, whether professionally or personally. So we're very pleased to hear what you have to say, Professor Jones. Thank you very much.
Thank you, Graham. Thanks for the invitation and for chairing today. Thanks also to Min and Valerie for organising this and for not inviting me to Cambridge. The train journey has pros and cons. I worry that nowadays PhD theses will never get read because academics are no longer taking long train journeys. And I was in the midst of time a Natski at Sydney Sussex College. So of course, I would happily seize upon any future excuse to revisit Cambridge. So I'm going to talk today about some long-standing, boring old background, some relatively recently published work, which you'd be forgiven for having missed, and then some more recent unpublished work, focusing particularly on the latter half of my talk on 22q deletion syndrome as a case study of psychiatric risk. So here's what life in my lab looks like. The lab is founded on in vivo electrophysiology, which means if you're a rodent, a rat on the left or a mouse on the right, you're likely to find yourself sporting a chronically implanted array of recording electrodes to monitor distributed neural activity throughout your brain with some focus on limbic cortical interactions, so hippocampus and prefrontal cortex in particular. You may have some stimulating electrode of some shape or form. So this rat has a little post on the back there where you laser light would be delivered to activate his midbrain dopamine neurons in this case. And you may or may not be genetically altered in some shape or form. We ally this rodent work to in vivo electrophysiology in patients and genotyped healthy participants. I can assure you and them there's no surgery or lasers involved in that case. And we rely on scalp EEG. So on the left here, is a participant sporting a relatively high density EEG cap, which he would wear through the night and through the subsequent day. And on the right is Amber, one of my PhD students, who's sporting a wearable EEG device, so in this case, a Dream headband, which has 6 EEG electrodes plus some accelerometry and pulse oximetry, for example. So the good news is the clunky technology is becoming more accessible and applicable. And then the analyses of neural activity that we apply to these different preparations, we try to use for integration. So using computational neuroscience, neural network models, for example, to interpret signals. And by and large, I'll spare you that today. We all know that the more equations you include in your talk, the less likely it is that anyone will be listening. So I'll focus on the in vivo phys, but I do think that there's lots of future mileage in computational psychiatry approaches. Now, one of the key advantages of this kind of approach, alongside its very high temporal and anatomical resolution, is that it is chronic, so we can record over extended timescales, both over the timescales of learning and remembering, so day by day, and over longer timescales as well. So in the case of the rat, for example, we monitor brain activity from individual animals for up to a year. It also means we can record activity whilst they're awake, doing some cognitive task of our bidding, for example, and during subsequent sleep. And it's this kind of approach that got me into the sleep field, although it's certainly not all we do in the lab. So as a postdoc at Massachusetts Institute of Technology, I was in Matt Wilson's lab doing this kind of stuff. So here's an example of a data set recorded from a single rat over the course of 3 hours, so time on the x-axis. During the first hour, this rat is drifting in and out of sleep. During the second hour, he's running around on a maze looking for chocolate. And during the third hour, he's falling back to sleep again. And the green line here is plotting population activity, so averaged hippocampal activity from a whole group of tens or hundreds of hippocampal neurons during the course of this 3-hour experiment. And you can immediately see that on average, the hippocampal activity during sleep after learning on the maze, so in this final hour of the recording, is higher than it was during learning itself. And this kind of notion was new to me. I'd blithely ignored sleep in my preceding neuroscience, and it really sort of alerted me to the neurophysiological capacity of sleep to support our waking behavior, if you like. So what's going on when we zoom into the details of these big spikes of activity in the hippocampus following learning? Well, that's what Matt Wilson is most famous for. And here's a summary of one 2002 paper from Matt Wilson's lab, performed by Albert Lee, who's now at Geneva Research Campus. So in this experiment, Albert was recording from hippocampal neurons of the rat. In this schematic, in this data figure, he's showing the activity of 10 neurons, numbered 1 to 10. And the colored dots here show the locations of firing of these neurons, so the location of their action potentials on a 2.5 meter long track. So the rat is running from left to right along this track, and these hippocampal neurons are place cells, Nobel Prize winning place cells as it happens, which means that they're firing clusters in particular locations in geographical space in this environment. So neuron 1, as denoted, happens to fire relatively early on a left to right run, whereas neuron 10 fires relatively late on a left to right run. And that sequence 1 to 10 is activated multiple times as the animal runs in this particular linear track environment. So Albert then continues to record from these same 10 neurons during subsequent sleep. And that sequence of activity 1 to 10 recurs more than you'd expect by chance. It's replayed during subsequent sleep. And that replay, shown here for a subset of the 10 neurons, again in that conserved sequence, coincides with high frequency bursts of oscillations recorded in the hippocampal local field potential, or EEG, essentially. The replay occurs on a compressed timescale. So although it takes the rat a few seconds to run 2.5 meters, this sequential replay during subsequent non-REM sleep occurs 8 to 10 times faster than that. And this prompts the sort of general model that patterns neuronal activity following learning is replayed multiple times during sleep at a time scale that's commensurate with the induction of synaptic plasticity, and that sleep-dependent replay process supports consolidation. So here's the cartoon model. We mooch around our worlds. As we do so, we induce patterns of neural activity that are contingent on the sensory input and the order in which it's received. So in this schematic, the rat activates place cell red, yellow, magenta, blue, and green in that order. And then those same place cells replay in that order multiple times during subsequent sleep, and that plays some part in consolidating memory. Now, obviously, that's a very superficial cartoonish view of what might be going on. For a start, a memory trace, an engram, doesn't reside in a single brain region, the hippocampus in this case. It's a distributed process. And memory consolidation isn't just about strengthening newly learned information. It's about integrating newly learned information into your existing knowledge base and perhaps prioritizing that information according to its salience. And here's one, two slides worth of example of what I mean. So in these experiments, same kind of deal. We're recording hippocampal activity from a rat. Here's a coronal rat brain shown on the left, denoting where our electrodes are implanted. We're also simultaneously recording from ventral striatum, so the nucleus accumbens of the same animals, whilst they perform a probabilistic learning task. So they have to decide which of 3 arms on a maze to go to according to their expectation of high, medium, or low probability reward. These experiments were run by Emma Roscoe, who was a PhD student in the lab at the time, and the analysis has been taken up by Tim, who's a postdoc in the lab. Emma is now a machine learning engineer. Such is the life of a modern neuroscientist. So if we examine the activity of ventral striatal or nucleus accumbens single neurons as animals approach the reward points at the end of the maze arms, we find that there's a signal denoting reward expectancy. So we've got 3 examples in the rows of this figure on the right-hand side of the slide of 3 different neurons. On average, they increase their firing on approach to the reward, which is at time 0. If we split trials into medium expected reward or high reward expectancy, we see that the firing rate ramps up more quickly on approach to a reward that's highly expected. So there's some kind of salience reward signal in those data. What's happening during subsequent sleep? So we continue recording from the same neurons in the same 2 regions during subsequent sleep. And we find that their activity is reactivated. So neurons engaged during this maze learning are more likely to be reactivated during subsequent sleep. And that's shown on this bar graph on the left. using a measure known as explained variance. I won't bore you with the details. Essentially, the bigger the difference between the blue and the gray bars, the more reactivation there is. And we're looking at reactivation of activity within the hippocampus, within the ventral striatum, nucleus accumbens, and across those two regions. And in all cases, there's more reactivation than you'd expect for neurons that are active on the maze. If we now relate that reactivation to the reward signaling, and we take the accumbens neurons, who are most strongly contributing to that reactivation signal during post-learning sleep, and we ask, what are those neurons doing on the maze? 3 different animals here. We find that the strongly reactivated neurons, shown by the blue traces here, are signaling approach to reward, whereas the neurons that aren't reactivated are not signaling approach to reward. So they're active in the task, but they're not signaling this, they're not carrying information about this reward signal. So this is just one example from my own lab, one of many from across the field, showing that reactivation of structural neural activity occurs during the hippocampus during non-REM sleep, but it coincides with information in other brain regions, including reward signals in the midbrain and other signals in the frontal cortical systems, for example, that reflects this distributed information processing during sleep. So from my very naive, non-clinically trained perspective, there's lots of scope here were this kind of process to go awry for a contribution to psychiatric signals. If you think about the process of memory consolidation as integrating newly learned information in a way that's somehow weighted or prioritized into existing knowledge, and if you imagine that that process has gone awry, then you could get, for example, patients dwelling on negative feedback, unable to let go of negative experiences. You could get patients inappropriately assigning salience to otherwise what shouldn't be positive feedback, for example. Hopefully you understand what I'm getting at. This kind of process gone awry could make contributions to symptoms and quality of life in patients. But how can we assay this kind of thing in those patient populations? We can't go about shoving electrodes very often into their hippocampus and ventral striatum. Well, the good news is that the nature of brain activity during sleep is highly conserved, and we can glean a lot of information from signal recorded on the surface using scalp EEG, for example, about what's going on in deeper brain structures. So if we take the case of non-REM sleep, we have cortical slow waves during stage N3 sleep. They are coordinated with thalamocortical spindles during N2 and N3 sleep. And in turn, those cortical and thalamocortical signatures are coordinated with hippocampal ripples. So in principle, taking a healthy human and using a non-invasive measure based on scalp EEG, one can at least make relatively direct inferences about what's going on in these other limbic and striatal systems. Here's another view of the coordination between these different sleep signals. And this is taken from data with intracranial electrodes in epilepsy patients, which means that we are, in this case, able to measure hippocampal activity. So what's being plotted here is the timing of a cortical slow wave during non-REM sleep. So here's a kind of grand average of a cortical slow wave in the top panel. Here's the relative likelihood of a thalamocortical spindle relative to the phase of that cortical slow wave. And here's the likelihood of a hippocampal ripple, this fast burst of oscillation that correlates with structured replay of activity, relative again to that slow wave event. So you see that there is an interrelationship. The phase of the slow wave shown on the top modulates the likelihood of a thalamocortical spindle or a hippocampal ripple. I do apologize, somebody's just started cutting down a tree outside my window. So much for staying at home to keep it quiet. Now, importantly for people like me, this kind of interrelationship is highly conserved across mammals. So here's an equivalent analysis from rat data, again showing that the cortical slow wave phase modulates the likelihood of a thalamocortical spindle or a hippocampal ripple. So this prompts a sort of general model of systems memory consolidation, whereby newly or recently encoded information in the hippocampus is replayed coincident with these fast hippocampal ripple events. That's coincident with thalamocortical spindle oscillations and cortical slow wave oscillations, with the latter acting as some kind of master clock or metronome, if you like, synchronizing limbic and thalamic and striatal activity during post-learning sleep to support this process of strengthening and embedding of knowledge during memory consolidation. So that, the humble EEG electrode allows us to infer quite a lot about the cellular and synaptic processes going on underneath it. And hence, the noble art of polysomnography has a lot to contribute to systems neuroscience. Here's Ulrich Bach, a grumpy German who was a postdoc in the lab, has recently set up his own lab at the Surrey Sleep Research Centre, who really sort of pushed forward the human EEG work in my lab. And of course, it's EEG work which allows us to characterize and score sleep and pull out sleep cycles, which in a healthy human iterate from non-REM to REM stages 4 or 5 times during the course of the night. And those stages are hallmarked by just different oscillations. So in non-REM sleep, the deepest stage of sleep is N3, and that's where slow waves come to the fore, these once per second oscillations potentially acting as a kind of master clock in the system's consolidation context. In stage 2 and 3 sleep, there are sleep spindles, which are faster, 12 to 15 hertz oscillation, reflecting reverberant thalamocortical activity. And both these things are coordinated with the faster, again, hippocampal ripple activity during non-REM. So these relatively coarse signals are positioned to tell us quite a lot. And here's one interesting example of that. If you consider the slow wave and how it's generated, here's a single slow wave shown in the middle of the left-hand panel on the slide. This slow wave at the EEG level reflects coordinated cellular activity underlying those electrodes. So as cortical pyramidal cells step in a synchronous fashion from depolarized up states when they're firing lots of action potentials or spikes to hyperpolarized down states where they're silent, as long as the population is coordinated and that's happening consistently, that stepping from one state to another about once per second will give rise to the slow wave of the EEG. So imagine then that your cortical pyramidal cells are highly interconnected with one another. That means that their steps are likely to be more synchronous. They're communicating with one another, co-driving one another. And so as they step from an excited high activity state to a silent low activity state, and those steps are synchronized, that will give you a steeper gradient of your slow wave. In contrast, on the right-hand panel here, if there's low synaptic strength interconnecting your cortical pyramidal cells, then they will act less synchronously. They're less well positioned to drive activity changes amongst one another, and so you'll get lower gradient slow waves. Now, these data are taken from a computational model from Giulio Tononi's lab, but if you apply them to the context of adolescence, then the inferences of the model start to gain some traction. So as I'm sure you know, adolescence is associated with synaptic pruning. So the exuberance of synapses generated in early life are then pruned away laterally, later in the frontal cortex during adolescence. And there's a refinement and reconfiguration of the neural networks as a consequence. So if you measure slow waves at different developmental stages, and these data are taken from 8 to 11-year-olds in the black bars versus 12 to 16-year-olds in the open bars, you see a decrease in the slow wave gradient. So this is reflecting synaptic pruning, giving rise to less strongly interconnected pyramidal cell networks, hence less synchronized during slow wave, hence lower gradients in sleep EEG slow waves. So in principle, this gives you a nice neurophysiological metric of the synaptic status of the networks underlying your electrodes. Something similar applies to thalamocortical spindles. So on the top panel on the left-hand side here, here's what a spindle looks like in sleep EEG in the raw data. Not very exciting to people who don't spend their lives obsessing about wiggly lines, but very gratifying for people like me. So we apply filters and detection algorithms to pull out the spindle events, which have different properties. There are slower spindles, which predominate over frontal cortices, and faster spindles, which predominate more centrally. Now, these spindles reflect reverberant activity triggered by the GABAergic inhibitory populations of the reticular thalamus, which then silence the thalamocortical projections. There's then a rebound activity following that silencing, which kicks the cortex into action, which then feeds back to the thalamus to complete the loop. So this reverberant cycle gives rise, the time constants of that reverberant cycle give rise to the 12 to 16 hertz spindle oscillation during non-REM sleep. So if you're measuring those oscillations and you see some change, then you're well positioned to make some inference about the mechanistic nature of that change at a cellular and circuit level. And because these mechanisms are so highly conserved across mammalian species, in principle, then you can go and test those predictions in animals, for example. Now, spindles came to the fore some time ago in the context of chronic schizophrenia patients, thanks in part to the work of Fabio Ferrarelli, again, working with Julia Tononi in Wisconsin at the time. And here's the paper that, alongside the hippocampal replay work, really sort of drew my attention to sleep just at the time I was setting up the lab, and led us to scrutinize sleep in a bit more detail. So here we're looking at a power spectrum of non-REM sleep EEG. So frequency is on the x-axis, and the amplitude or power of oscillations at those frequencies are on the y-axis. You see there's a lot of action going in at slow frequencies corresponding to the slow waves of non-REM sleep. So that's the first peak of the power spectrum. But in healthy subjects in blue, and in this case, depression patients in green, there's also a lump, there's also a peak around this sort of 12 to 15 hertz frequency, which corresponds to the sleep spindles. And in this particular study, in chronic medicated schizophrenia patients, there's an attenuation of this hump. So on the face of it, that's an interesting signal to start exploring in a bit more detail, right? In principle, were it specific in any way, then the blunting of sleep spindle activity in schizophrenia patients might give you some clues that there's, for example, thalamocortical function in those patients. Perhaps that thalamocortical dysfunction would manifest during wake as intentional impairments, for example, and so on. So you can start to piece together a sort of translational narrative. This raises lots of questions, of course. These patients are on a mixed bag of medications. The result has been repeated by us and others. So here's an example from some of our own work, analyzing a different data set, again showing healthy participants in blue with a very robust spindle peak at 12 to 15 hertz, and an attenuation of that peak in schizophrenia patients in this case. But again, they're adults, they're chronic patients, they're on a mixed bag of medications. In principle, just being told that I've diagnosed you with schizophrenia could affect your sleep spindles. In our defense, though, there's evidence showing that first degree relatives also harbor this marker of abnormal activity during non-REM sleep. So, all is not lost. And I think there's still more to be gained from understanding the mechanisms giving rise to this abnormality, and or whether it's specific to schizophrenia or not. And there's a lot of debate about that. Some say it is. These data on the left suggest it might be, but others have shown spindle abnormalities in patients with major depressive disorder, for example. So we took these data and back translated them into the rat. This was some time ago now. We used the rat model of neurodevelopmental disruption. Of course, it's not a model of schizophrenia, but the idea was to briefly interfere with embryonic development of limbic cortical circuitry, and then see as animals matured whether that gave rise to any phenotypes reminiscent of the clinical scenario. And in non-REM neurophysiology terms, it did. So the clunky bar graph on the left here shows a reduction in sleep spindle density in the model rats as opposed to the sham control rats in gray. But if we scrutinize the signals in a bit more detail and ask, okay, what about the coordination of activity across different parts of the brain? So how coordinated is that activity recorded at the front versus recorded at the back, for example? Again, we see an impairment in this particular model. So the gray line here shows the degree of coordination of slow waves during non-REM sleep across frontal and posterior EEG sites in the rat. And the blue line shows that the degree of coordination is quite markedly attenuated in these models whose neurodevelopment has been disrupted 3 or 4 months before these recordings were made. There's also a discoordination of slow waves and spindles in this particular model. So having gone from someone else's clinical data to back translation into rat, we then cycled back to clinical data in collaboration with Dara Manoak, who's at MGH. And she had an extant sleep EEG data set from schizophrenia patients. The behavioral context of these data is shown here. So patients are brought into the lab. They have a baseline night of recording. So their first night of recording of sleep EEG. Then they're trained in a motor sequence task, the MST. So they're asked to type a sequence of digits as quickly and accurately as possible with their non-dominant hand. And they're given 12 training trials, shown on the left-hand part of this plot here. And patients in red and healthy participants in blue both show a training-dependent increase in performance. There's then a second night, again coupled with polysomnography to record sleep EEG, and then a morning testing session. And you see that healthy participants show an overnight improvement in their performance in the task. So their performance in the morning is significantly better than it was at the end of training the evening before, whereas patients do not show that overnight improvement. So our question was, can we predict this behavioral impairment on the basis of sleep EEG? And we published a couple of different studies over the last few years showing that our ability to predict the degree of overnight improvement in this motor sequence task is blunted in schizophrenia patients as opposed to healthy controls. So what we're plotting on these correlations here is the amount of improvement in the motor sequence task performance versus our prediction of how much it will improve based on analyses of sleep EEG. And for this admittedly small set of healthy controls, most sit on the nice straight diagonal, whereas there's much more scatter and variance in the schizophrenia patients. But again, we're at the mercy of a mixed bag of medications and very mixed histories in these outpatients in this case. So we wanted to start to tackle the ugly question of chicken and egg a little bit. And we did that in Bristol by taking advantage of ALSPAC, the Avon Longitudinal Study of Parents and Children, otherwise known as Children of the 90s, which is a birth cohort of 10,000 kids or so who are now in their early 30s and have been under the microscope since birth, and they're genotyped. So we undertook a recall by genotype study based on the single locus ZNF804A. Sorry, there's an errant A there, it's just ZNF804A, which was one of the first risk loci to be identified in genome-wide association studies for schizophrenia. It's an enigmatic protein, so the gene encodes a transcription factor. It's highly expressed very early in development. It's expressed in the fetal populations that give rise to cortical interneurons, for example, and it's positioned to modulate synaptic plasticity, although the data are somewhat enigmatic. We picked this locus partly because we had a null mutant mouse as well, so there's a sort of allied set of mouse analyses I'm going to talk about today. But really it allowed us to study or ask whether there was any association between abnormal sleep-dependent behavior and sleep-dependent neurophysiology in an otherwise healthy population. Because of course, this is very low penetrance risk variant, it only increases your risk of diagnosis by 1.5% or something of that order. So we used the same approach. Participants brought into the sleep lab, they're trained in the motor sequence task, they're recorded overnight in the sleep lab, and then we ask how well can we predict overnight improvements in performance in the behavioral task using our suite of neurophysiological metrics. On the left here, we're looking at slow wave activity on the pre-training night, night 1, and the post-training night, night 2. And in this particular participant who's homozygous for the non-risk variant of ZNF804A, you see that frontal slow waves in the pre-training night are not particularly aligned with posterior slow waves under those sort of baseline conditions. But when we do the same analysis post-training, then frontal slow waves, shown at the top, do appear to coincide with slow waves recorded more posteriorly. These are more posterior EEG electrodes. So if we take metrics like this and develop a kind of composite neurophysiological metric of behavioral performance, this is how well we can do in black in panel C here in the participants who are homozygote for the non-risk variants of the CC group. And again, we can do a fairly good job of predicting behavior based on the neurophysiology in this group. Whereas in the group that are homozygous for the risk variant of ZNF804A, despite the fact that they're perfectly healthy and at very low risk of schizophrenia on the basis of this one polymorphism, then we actually can't predict how well they're going to do or how much their behavior is going to change overnight. So it's only got as far as a tantalizing result, I guess, but again, it lends to the evidence that abnormal sleep neurophysiology and sleep-dependent behavior in schizophrenia patients isn't a kind of epiphenomenon or a side effect of medication, for example, that perhaps it's a mechanistically insightful biomarker, and perhaps it could be used as some sort of predictive biomarker as well. So to test that in the last few slides of my talk, we moved to 22q11.2 deletion syndrome, sometimes known as DeGeorge syndrome. It's a copy number variant, so it's a chromosomal deletion syndrome that typically affects around 40 to 50 genes. So carriers of the deletion of the microdeletion are heterozygotes for 40 or 50 genes. Because there's a number of genes involved, presumably it's a complex syndrome. It doesn't only affect the nervous system. There are craniofacial abnormalities. There are cardiovascular abnormalities, and there's a lot of heterogeneity in deletion carriers. So, it's not cut and dried straightforward, but it's clearly an interesting case study because, on average, about 1 in 4 young people who carry the deletion will go on to develop schizophrenia. A greater proportion will develop some other psychiatric diagnosis. And there's also a strong association with reduced IQ, for example. Now, this study has been enabled by our collaborator in Cardiff, Marianne van den Brie, who's an equal partner in all of this. And Marianne has spent a lot of time curating and developing and liaising with a cohort of 22q deletion syndrome carriers and their families, along with some other copy number variants cohorts as well. It helps to be in the capital city of Cardiff, of course, because all the patients coming through can be genotyped. The work was undertaken by Nick Donnelly, who's an alumnus of Cambridge, who I actually met when I was examining his PhD in the Department of Psychiatry, although most of his PhD was done with Jeff Daly in psychology at the time. And Nick has just been appointed an academic clinical lecturer in psychiatry in Bristol, I'm pleased to say. And Ulrich was again to the fore here, who's recently set up his lab in Surrey. Because the deletion syndrome is quite rare, 1 in every 2,000 to 4,000 live births, the patients are spread around the country. So we pack our EEG kits in our bag and we go to their homes in this case. So we're not bringing them into the sleep lab. That may be important, of course. And instead of a motor sequence task in this case, we're using a sort of paired association task, a sort of matching pairs game where participants are shown matching pairs on the screen during training. And then during testing, they're shown one of the two and asked to identify where the other one is. This was in part to invoke hippocampal processing more directly, and in part because they really like it. They really like playing on the iPads. So we do that before sleep. We record polysomnography overnight, and then they wear the EEG kit the following day as well. So it's a 24-hour recording, not a 7-day recording. And that was in part to capture seizure-related activity, which is also evident in this population. Again, one motivation for starting with this particular copy number variant is that there are mouse models. But again, I will spare you the mouse work today. I'm happy to answer questions. So our question was, can we detect any phenotypes at the behavioral and or neurophysiological level in these kids prior to any frank psychiatric impairment or diagnosis that's reminiscent of the phenotypes we see in schizophrenia patients? As expected, performance of these kids on the task is impaired. And so it takes the 22q carriers, shown in blue here, more training cycles to get to asymptotic performance than it does their unaffected siblings, who are shown in gray. And when we test them the following morning, the 22q carriers make more errors than their siblings. That's parsimonious given reduced IQ in the syndrome. Sleep is largely intact, but there are some disruptions based on these polysomnographic measures. So on the right here, we're looking at a polysomnogram throughout the course of one night of a a sibling compared to a deletion carrier on the bottom. So, the sleep cycles are all there. They're largely structured as you might expect. But in previous questionnaire-based studies in these kids, we had shown that 60% of them have sleep problems, particularly sleep fragmentation and some degree of insomnia. And at a sleep architectural level, there's a slight increase in the proportion of stage 3, so slow wave non-REM sleep. In the deletion carriers, a slight decrease in stage N1 and a slight decrease in REM sleep as well. But of course, most of the interesting phenotypes we'd seen in adult patients had required us to dig into the details. And so Nick and Uli went in to detect individual events, starting with slow waves. And so what we're looking at here is a kind of grand average of slow waves recorded on a single electrode from the siblings and the deletion carriers in blue again. And you can see that the slow waves tend to be a slightly higher amplitude in the deletion carriers. On the bottom here, that's plotted topographically. So this is showing in sensor space, in EEG electrode space, where those high amplitude slow waves were. But if you think back to my case study of the neurodevelopmental profile of slow waves, so as the adolescent frontal cortex matures, then the slow waves become less steep and potentially lower amplitude because of synaptic pruning and less interconnected populations underlying them. Then what we might be picking up here is effectively a sort of neurodevelopmental effect. So if we plot slow wave gradient versus age in this cross-sectional study for the typically developing siblings on the top versus the 22q deletion carriers on the bottom, we see the expected age-dependent decrease in slow wave gradient in the siblings, and that's flattened and sitting at a slightly different level in the deletion carriers. So that's kind of good and bad news. On the one hand, it's an interesting neurophysiological metric of neurodevelopmental status. On the other hand, without a bigger cohort and a longitudinal study, it becomes quite hard to unpick sort of grand average effects like this one, because it might just reflect variance in developmental rate, basically. What about the spindle activity that's been shown multiple times to be attenuated in schizophrenia patients and their first-degree relatives? Well, that does also appear to be attenuated in 22q deletion carriers, despite the fact that these are relatively young people. Sorry, I forgot to dwell on the age range of this study, but we've got quite a broad range of participant ages from 6 to 20 years. The median ages of the two groups, the siblings and the carriers, are the same, around 10 years, I think. So these kids haven't yet shown, with the exception of 1 or 2 cases, any frank signs of psychosis, for example. All the more reason to keep studying them. Nevertheless, there is a blunting of spindle activity. So the carriers are in blue and the siblings are in gray. Again, you see that sort of slight reduction in the spindle peak. That's not the same as, but at least somewhat reminiscent of the blunting of spindle activity in patients, adult patients shown in red, and versus healthy participants shown in blue in this reminder plot from an earlier slide. So, obviously one of the things we'd like to do is follow these kids, see whether, those kids with the most significantly affected slow waves and spindles, for instance, are those that turn out to be most at risk in clinical terms. In terms of relating the neurophysiology to clinical symptoms, then Nick ran a mediation analysis, and it seems to be that the effects on slow wave amplitude of 22q deletion are mediated by ADHD symptoms in particular. So on these plots, we're plotting in the white circles the electrodes where there's a significant mediation effect, and almost all the significant mediation effects relate to ADHD symptom-mediated impact on slow wave amplitude. And this is kind of interesting because in our questionnaire-based studies, we'd found that restless sleep in 22q children was associated to ADHD symptoms in particular. And in longer-term studies run by Marianne van den Brie and her team, early ADHD symptoms do appear to presage later psychotic symptoms in the longitudinal studies they've done to date. So it may be that we're able to relate these neurophysiological signatures to the sort psychopathology dynamics, I guess, as kids progress from childhood to adolescence. So a kind of interim conclusion, but don't worry, it's very close to the end. What I'm trying to say is that sleep is conserved. That's useful from a translational perspective. We all know it's vital, and it's disrupted in some shape or form in almost every mental illness. We do have to be very careful though, because sleep can only be properly interpreted in its 24-hour context. And as an example of that, we've trained people in lab-based tasks and then related their behavior, or shown that that experience does impact subsequent sleep physiology. So if you just study sleep physiology and knew nothing about what was going on in your patients during the day, it would be very hard to interpret. Of course, in populations like schizophrenia patients, for example, who tend to be up all night and sleeping during the day, there are key interactions to unpick there. And we need to be careful about the use of the sleep lab as well, potentially. It may be that by dragging patients into a sleep lab, you unintentionally kind of realign their circadian rhythm and sleep rhythms, and therefore miss some of what's actually going on in the field, if you like. Nevertheless, I do believe that non-REM sleep neurophysiology is a useful translational biomarker. It gives us mechanistic insight and allows us to make predictions that could be tested in animal and computational models. And in the context of 22q deletion syndrome, it may be that some of these slow wave and spindle abnormalities presage subsequent problems as children mature and transition through adolescence. But all those long and expensive longitudinal studies are what's required to tell us that. Importantly, what I'm not trying to say is that sleep is always to blame. As a sleep researcher, it's easy to start thinking that. And I'm not saying that sleep disruption necessarily causes schizophrenia. I think what I'm really trying to champion is the use of these sleep-based metrics as mechanistic biomarkers. It's not necessarily the case that reduced spindles is causing schizophrenia, for example. So very quickly, before I finish, where do we go from here? Well, one obvious next step would be to try and fix it. So if you have a sleep or an abnormality that manifests during sleep, one might try to use drugs to fix it. And we've done some recent work in rats, recording simultaneously from hippocampus, frontal cortex, and other regions in striatum and amygdala, for example, looking at the effects of some sleep drugs. Benzodiazepines, no surprises there, generally mess up coordinated activity during sleep. So from a kind of sleep-dependent cognition perspective, they're likely to exacerbate problems. Whereas some of the Z drugs at the appropriate dose can augment coordinated activity during sleep. And we're looking here at the effects of zolpidem in the rat on coordinated interactions between the hippocampus and the frontal cortex, which can be enhanced by low doses of that drug. Now, dose equivalence is very difficult to figure out, but maybe all is not lost for some of the drugs that are already prescribed. Another more direct approach is to use some kind of stimulation, of course, and for quite some time now, people have been pondering the applicability of transcranial stimulation to modulating brain activity during sleep. So here's a 2006 Nature paper from Jan Born's lab using TMS to induce slow wave activity during non-REM sleep, showing in healthy participants in this bar graph in B that augmenting slow wave activity during sleep improves memory. So in this case, the number of word pairs that can be recalled the next day. And neurophysiologically showing that slow wave stimulation, so at this 1 cycle per second rate, also augments spindle activity. So the stimulated group in this case is in red, the unstimulated controls are in black. And the slow wave stimulation, which was down at these 1 Hertz frequencies, has augmented spindle activity. And again, a little reminder graph, if you think back to the Ferrarelli study showing that the spindle activity was blunted in patients, then of course it's tempting to speculate that some sort of stimulation intervention would be worth testing there. And that's beginning to happen. A final comment is that having raised some caution about the sleep lab, luckily we don't need to use it anymore. As I said, our study in 22q was done in people's homes, partly born out of necessity. But the tech makes it more applicable nowadays. And again, here's Amber wearing a Dream headband device. So perhaps we can start to capture sleep EEG data in the field at scales that's more applicable to interrelationships with GWAS studies, for example. And I'll spare you the details, but this study from our collaborator, Sean Purcell at Brigham and Women's, exemplifies that. He's curated a large sleep EEG database in the States, and 8,000 of these 11,000 individuals are genotyped. So I will end there by thanking my hosts in Bristol, the funders. Most of this work has been enabled by my Wellcome Senior Fellowship, which I'm very sorry to say is about to end and can never be rekindled. We collaborate with Lily, Compass and Boehringer on aspects of this work, and the 22q study just wouldn't have been possible without our neighbours across the bridge, and in particular Marianne van den Brie, who allowed us access to her precious patients. Thanks, of course, to my lab. So Uli, who led the establishment of the human work in the lab, is now in Surrey. Emma, who did the striatal work I talked about earlier, in particular, and Nick, who I mentioned, Nick Donnelly, who we're pleased to have established in Bristol nowadays. Thanks very much.
Well, thank you very much indeed, Professor Jones.